Hi, ladies and gentlemen. Today we're going to be talking about Chapter 20, Phylogeny. And the term phylogeny really just means evolutionary relationships of a species or a group of species. If you are a biologist and you are working in the discipline of systematics, then you are looking to classify organisms based on their evolutionary histories, and you're going to use evidence based on morphology and most notably biochemical or DNA evidence to help group organisms based on their history. And the term taxonomy really means the science of classifying and naming organisms, which brings us to the father of classification himself, who is Linnaeus. He lived in the 1700s, and he was the first one to really group organisms into the 7-layer classification system: kingdom, phylum, class, order, family, genus, species. Don't worry, I will give you an easy way to remember that because you do have to know it. But he also came up with the idea of binomial nomenclature. We're giving an organism a scientific name, which includes its genus and its species. The genus name is capitalized, the species name is not. Well, the first letter of the genus name is, and it's generally italicized. Notice this slide has several stars on it. It is very important. What it does not show, and Linnaeus did not come up with the domains, but we actually have 3 domains. Eukarya, which includes all eukaryotes. So if you have a nucleus, which is a lot of organisms and life on this planet, are one group. And then there are 2 different prokaryotic groups, which are archaea and bacteria. And they are different based on some of the structures in their cell walls. So that would be really on top of kingdom, and it would be the most inclusive. But kingdom on this chart includes the most number of organisms, and a kingdom would be made up of many phylum. A phylum would be made up of many classes. A class is made up of many orders. An order is made up of many families. A family contains many genera, so several we don't say genuses, we would say genera. However, when you get down to the species level, it is one organism, period. So this chart shows the classification of us, human beings, Homo sapiens, and we are in the genus Homo. We are sapiens. That describes who we are. There has been identified a distant cousin or relative of human beings called Homo erectus. They were in the same genus as we were, but their species name is different, such that had we gotten together, we would not have been able to make fertile, viable, or offspring that were fertile. We could have made viable offspring, who knows, but they wouldn't have been fertile by the biological definition of the species. So here's a picture of a phylogenetic tree with just a couple of things to point out. This would be considered a clade. All of this would be a phylogenetic tree. And we can see here some relationships between A, B, and C. Because B and C share a common ancestor, and we don't know anything about that ancestor, but because their ancestor is more recent than the ancestor to A, we can say B and C are more closely related. Question I have for you when you look at this chart is, who's more closely related to A, B, or C? Now, your initial gut reaction would be to say, oh, well, B is more closely related because look, it's listed right next to it in the chart. But if we took that B and C loop, we could actually rotate it right here on the node. Okay, we can rotate it and we could list it as A, C, and B, and it doesn't change the relationships because no matter how we line it up, the most recent common ancestor that B and C share to A is way back there, and it's equivocal. So we can say without a doubt that B and C are more closely related because they share a common ancestor here, and they're the only 2 that branch off of it. But neither B nor C is more closely related to A based on this tree. So don't fall into the trap of the order of things indicating relationships. Okay, so I believe you had an activity where you were charged with creating a phylogenetic tree for 4 groups, A, B, C, and D, excuse me, where C and D are more closely related to one another. And then C and D together are more closely related to B, and then A is sort of an outgroup. And if you came up with something that looks like either A or B, you're spot on. They are the exact same tree, indicating the exact same information, even though they look a little bit different. So the directions were C and D are more closely related. That is true based on the representation of both of these trees. And it also said that C and D are more closely related to B. So we have them together with their common ancestor and more closely related to B. Perfect. And they are more closely related to B than A. And so we've got A here as this outgroup. It's all by itself. It doesn't have a little clade of its own. It's all by itself. In both of the trees, A is an outgroup. In fact, in all three of the trees, A is an outgroup. This tree is incorrect because if you look at it, C and B are more closely related, and then C and B are more closely related to D than A, and that is wrong. That is not what the instructions ask for. So what we can and cannot learn from phylogenetic trees, they basically just show us patterns of descent. They do not indicate exactly when speciation events occurred. They do not indicate exactly how much genetic change has occurred over the time. And we certainly shouldn't assume the order of the taxon is an indication of relationships. Once again, B is not more closely related to A than C or D, because B, C, and D all share the same most recent common ancestor with A. Okay, so don't get caught up in the order. Okay, excuse me. These phylogenies are inferred from looking at data. It used to be morphology or body shape was the main tool used to classify organisms. Well, if you ever played the game Uno, the best card to have when you're left with one card and you call Uno would be the card that has 4 colors on it that you can play at any time, no matter what card is being laid down right before your turn, because then you're like, boom, won the game. And in that kind of situation, we would call that a trump card, something that beats all else. I'm using that analogy because DNA sequences are literally the trump card for phylogeny. Similar morphologies can be the result of convergent evolution. Similar morphologies can be the result of similar environmental pressures selecting for a certain phenotype that is more successful in that environment. So really, the end-all be-all piece of evidence for phylogeny or even determining relatedness would be biochemical evidence, i.e. DNA. A couple terms here to go over. Homology. If we have homologous structures, that means homo means same. That means they are the literal same structures, and that indicates shared ancestry. We talked about this in chapter 19 when we were talking about evidence of evolution with the mammalian forearm. All of the bones in my forearm are present in every single mammal. They might have different shapes or different sizes, but they are present, and that indicates shared ancestry or common descent. Analogous structures, or an analogy, is a similarity due to convergent evolution. So let's take the shark and the dolphin, and let's take their pectoral fins, the ones that that are kind of in the front. They both have a similar body shape because as a top predator, that shape has an advantage in an aquatic marine environment. If we were to examine the structure of those pectoral fins, we would find all of the bones from the mammalian forearm in the dolphin's pectoral fin. We would not see bones in the shark's fin because it is not a bony fish. It's a cartilaginous fish. And the tissues and the origins of those structures are totally different. They are analogous to one another because they are the product of convergent evolution. Couple more vocab words right here. Shared ancestral character and a shared derived character. I think it's easier to look at this diagram when we talk about these. If we examine the lamprey, the tuna, the salamander, the turtle, and the leopard, and I asked you, of these characteristics or traits, which one is a shared ancestral trait among the group? Which one is shared and it evolved a long time ago? It would be the vertebral column. If I asked you between the turtle and the leopard, what would be a shared derived characteristic or a shared derived trait? That would be the amniotic egg. You couldn't say hair because only the leopard has hair and it would not be shared. Okay, so derived means more recently evolved trait, and ancestral means evolved a long time ago. Okay, one of the cool things scientists can do with phylogenetic trees and phylogeny in general is that they can predict features of an ancestor based on the features of its descendants. And so we've got a phylogenetic tree right here of reptiles and birds. Obviously, the dinosaurs are no longer living, but we've got crocodiles and birds that are living and are both the descendants of some shared common ancestor that we really don't know what it is, but it's way back here. If we look at some of the things birds and crocodiles do, we see some similarities. We see brooding behavior where they build nests and lay eggs. And we also see some parental care here. This crocodile is not eating its baby. It is being a good crocodile mommy and taking care of it. So based on that, what could we predict about this ancestor if we see both of these behaviors and traits in current living descendants? It isn't really a big stretch to predict, and we have really no way to absolutely confirm that this ancestor probably had brood behavior and maybe even some paternal care. Now, I'm not saying at all that this is the lost ancestor of reptiles, crocodiles, and birds, but fossilized dinosaurs have been found sitting on eggs in exhibiting brood behavior. Not rude behavior. You can see the eggs here, and you can see the mama crocodile or mama dinosaur sitting on them. So I think it's kind of interesting that we can apply some stuff sort of backwards and make predictions. Now, we have really no way to confirm it without seeing absolute evidence, but it's interesting that we can extrapolate what we know and apply it backwards. Alrighty, an organism's evolutionary history is documented in its genome. That is for sure. When we compare nucleic acids and other molecules, we can infer relatedness, and it's a really good tool for tracing evolutionary history. And scientists have learned to look at different genes because different genes evolve at different rates, or different genes change at different rates. So for example, DNA that codes for ribosomal RNA, that is a gene. It does not code for a protein. It stops at the RNA level. Our RNA makes up our ribosomes. Ribosomes are the site of protein synthesis. If there is a mutation in the gene that codes for our RNA and it makes non-functional ribosomes, what do you think is going to happen to that organism? Yeah, that organism is totally going to die. That organism is not going to live, and it's going to take its crappy DNA with it. So our RNA changes very, very slowly, and the changes that have accumulated over time have been changes that are tolerable and that do not damage the function of a ribosome. Our RNA would be considered highly conserved genetic code. On the other hand, mitochondrial DNA is DNA found in the mitochondria, not nuclear DNA, because remember, we believe that mitochondria and chloroplast both used to be prokaryotes billions of years ago, and so they have their own DNA. Mitochondrial DNA does not control every biochemical reaction of the cell, so changes in mitochondrial DNA can be tolerated, and it actually evolves quite rapidly and can be used to explore recent evolutionary events. Okay, that is the end of the lesson for chapter 20. It was very short. I recommend that you go through the review questions on the posted PowerPoint and let me know if you have any questions.